Hi, today I'm going to tell you about the Chemistry of Life pH and Buffers lab. I'm going to walk you through the instructions, and then I'm going to show you some of the equipment and materials you'll be using to do the lab. I won't show you the results, but I am going to give you some detailed information because this is a fairly complex lab this week. So if you have the correct lab, you'll see the frog on the first page, Chemistry of Life pH and Buffers. This first part just talks about pH and buffers, and you have already learned about pH and buffers in the properties of water lectures. So if you'll recall, pH scale is 0 to 14, with pH 7 being considered neutral. Any pH below 7 is considered an acid, and above 7 is considered a base. Also remember that regulation of pH in a living organism is extremely important because when pH changes dramatically, it really affects protein structure. It can denature proteins. And in particular, we're worried about enzymes that carry out key chemical reactions in a living organism. All proteins can be affected though by changes in pH. And that's why we have buffers. Remember that buffers serve to keep pH relatively constant, not pH 7. Remember that all areas of the body do not have the same pH. So even just looking at the human body, the pH in your stomach is dramatically different from the pH of your bloodstream. So buffers serve to keep pH normal, whatever normal is for that system. And remember that buffers work by either tying up acid or adding more acid or adding hydroxide ions. So adding hydroxide ions would bring the pH up. Adding hydrogen ions would bring the pH down. Tying up hydrogen ion could also be a way to bring pH up. And that's what happened with the carbonic acid buffer system that we talked about in the properties of water lectures. If you look at this time requirement, I would say that this preparation of 30 minutes is actually a big understatement because the lab is complex this week. And really, when I go through a lab activity for the first time, I like to highlight the first round, and then I like to read the whole thing again and really write down, take organized notes for the steps that I'm going to be carrying out. There is a lot going on in this lab, and so I would budget about an hour for prep, not only for reviewing it, understanding what you're doing, but there's going to be some setup and labeling that you're going to have to do before you start the activities. I also want to note that you're actually doing 3 activities in this lab. So activities 1 and 2 are in the instructions with the frog. The third activity, the instructions for that are on the lab questions, and I'll show you those in a minute. You do need all of your personal protection equipment. We're working with a strong acid, hydrochloric acid, and a strong base, sodium hydroxide. It's very important that you wear closed-toed shoes, that you wear the lab apron that's provided to you. I have my lab coat on, but you really probably want something waterproof as well. You want to wear your gloves and you want to wear your goggles. So in your bag that has your chemistry safety kit, you have all of those things. You have your goggles. Okay, your eyes are very important. Don't take a chance. This strong acid and strong base today can really damage your eyes. So wear your goggles, wear your gloves. Have your camera ready because you're going to be taking several photos for parts 1, parts 2, and parts 3 of the lab today. Please don't dispose of anything until you have taken pictures. I don't want you to make a mistake and dispose of your first activity results and then rinse it out and realize, oh no, I didn't take a picture. Make sure you take pictures at the end of activity 1, at the end of activity 2, and then for activity 3, there are 2 different photos you're going to take. So you're gonna have a total of 4 photos this week for your lab. Okay, there's a lot of background about pH here. You can skim through this. The directions say that you need a calculator today to calculate logarithms. That is not true. You do not need to calculate logarithms as part of the lab today. Remember, we ordered these lab kits. This is a chemistry lab. And for this class, you don't need to worry about the logarithms. So you can just skip over that part. There are not any questions about that in the follow-up questions about the lab. So don't worry about the logarithm part. You will need a calculator today, though, to calculate some molarity and dilution factors that I'm going to show you in a minute. Okay, the other thing you don't need to worry about is this Henderson-Hasselbalch equation. Again, this is for a chemistry class, so you can ignore that part. Otherwise, you can just read about buffers, which you should already know about. Okay, this is where you need to really focus though, dilutions. This term molarity, if you recall from the properties of water lecture, this is how we measure the strength of a solute. So remember, a solution is a solute plus a solvent. That solvent is usually water. And in this case, we're going to be diluting acid and base with water to get to a specific strength. And the way we measure that strength is molarity. This calculation is showing you that you can use ratios to determine how much water needs to be added to get a specific strength of solution. So this is saying molarity initial and volume initial equals molarity final times volume final. I think I said that wrong the first time. Let's say it again. Okay, molarity initial times volume initial equals molarity final times volume final. I actually like using ones and twos here. So I like saying molarity 1 times volume 1 equals molarity 2 times volume 2. So what you'll do is read this example, okay, and actually in the lab questions today, they're going to ask you to do this exact same calculation. But for the experiment, the calculation you're going to be doing is different. So I have actually, I'll show you this again when we start actually doing the experiment, but I actually wrote the calculation that you need to do today. It's the same calculation twice. One time it's doing the same calculation with sodium hydroxide, and the next time it's doing it for hydrochloric acid, but the numbers are the same, so your answer is going to be the same. In the lab today, you're going to be looking at making a 0.1 molar solution, but you need 12 milliliters total of that solution instead of 10. And we'll talk about that more when I show you the exact lab activity. Okay, 3 bags of materials you're going to need today. One is your personal protection equipment. Okay, so that's your goggles, your gloves, your apron. Okay, then you need the materials that are labeled for the pH and buffers lab. That's all of these things. This is the tray where you're going to be labeling this tray and carrying out your experiment in this tray for activity 1 and for activity 2. So again, you want to make sure after doing activity 1, you're going to have different colors of solution in each one of these wells. Be sure to take a picture of that before you dispose of it. And then you're going to rinse it and you're going to use that same tray for activity 2. Okay, the other equipment you need today comes from the bag that is your general materials bag. Okay, so you're going to need a beaker, a graduated cylinder, 2 of these little dropper pipettes, 5 test tubes, a test tube rack. This is actually cardboard. You're gonna kind of just fold that. You'll see how that folds to make a test tube rack. And then you need your, what they call a grease pencil. It's actually a wax pencil. I would say if you have a Sharpie, you could use that too. It actually might work out better. You'll just have to somehow cross through that writing in the future. The nice thing about the wax pencil is you can rub it off, and we will be using some of this glassware again. Okay, needed but not supplied. Distilled water, you can use tap water for this lab. You do not need to go purchase distilled water. You can use tap water. Calculator, you do need a calculator to calculate your dilutions for the molarity part, but you do not need to calculate logarithms. So you can just use the calculator on your cell phone. You do need a camera to take pictures of your results, and obviously something to write with. The other thing you need is for activity 3, you need a drinking straw. So you can just go anywhere and get a free straw at Starbucks, at 7-Eleven, at McDonald's, wherever you can go and get a free straw. You need 1 drinking straw for activity 3. Okay, you are working with a strong acid today, hydrochloric acid, and a strong base, sodium hydroxide. These are both toxic. Do not chew gum while doing the lab. Do not eat or drink anything while you're doing the lab. Really make sure you wash your hands well after, even though you're wearing gloves. Please be very careful with these chemicals today. Also, the pH indicator that we're using today is flammable, so please be really careful when you're doing the lab. Okay, so the instructions today are fairly complex, but I just want to show you that all of the directions for activity 1 and activity 2 are in the frog document. And you're going to be recording your data here. But ultimately, you're going to transfer that data to the lab questions, and that is where the directions are for activity 3. Okay, so these are the lab questions. This is the submittal you're going to make this week for 30 points. And you'll see that all the questions are here for activity 1. These are the same calculations that are in the example of the lab exercise. You're going to, again, be doing a different calculation because you're going to be calculating it for 12 milliliters for the actual lab exercise. This is where you'll record your data. This is your explanation of what happened in activity 1. And again, be sure to take a picture because at the very end is where you're going to put your photos of all 3 parts of the experiment. So this says insert a photo of your results of activity 1. Okay, activity 2, completing the data table, answering the question, and then inserting your picture. And finally, activity 3, you need a drinking straw for this activity. You're going to fill a beaker with water, add the pH indicator, note the color, and take a picture. Then you're going to take the straw and you're going to blow bubbles for 2 minutes. So you can take breaths, but keep blowing as many bubbles as possible. Add pH indicator again, and now note what the color is. Think about what you're exhaling. What gas is that you're exhaling? Okay, so we breathe in oxygen, we exhale. I won't tell you, but you should know. And so think about what that chemical is that you're exhaling, and is that an acid? Is it a base? When you're discussing how the pH changed. Again, photos. So for part 3, you're going to have a photo of the beaker with the water and pH indicator before blowing bubbles. So that's your control, and then after blowing bubbles. Okay, now I'm going to show you some more details about the lab activity. I'm going to go into my kitchen. Okay, so here I am in my kitchen ready to do the experiment, and the prep today takes a little bit of time. So again, there are 2 bags besides just your safety equipment that you need to pull out. One is the one that says equipment set, and that has a bunch of equipment in it that you need today. Okay, and then the other one is the one that says chemistry of life, pH, and buffers. And I apologize, I already took things out of these bags, so. You just want to really lay everything out so you're really organized and ready to go. And also you can see that not only have I've highlighted, but I've also gone through and I've actually written detailed directions for what to do. This is a pretty complicated lab. So the preparation that they take you through goes through activities 1 and 2. Honestly, I like to do all the prep for activity 1 first, do the activity, and then go back and do the prep for activity 2, and then do the activity. So I'm just going to go through this. I know you guys can read, but there are some things I want to show you. First, you want to have your goggles on. So I'm going to put my goggles on right now. You can't see me, but I do have them on. And they are pretty loose when you first put them on, so make sure you tighten them so they actually do stay on your face. Which you just pull the tabs on the side, the elastic on the side. Okay. Now, the first thing it asks you to do is use a grease pencil to label 2 test tubes. So you're going to go into your general bag and you're going to pull out all 5 of the test tubes. And what I like to do is initially keep them in a beaker, but eventually you're going to be putting them into the test tube rack, which is this cardboard thing, which actually seems to work pretty well for cardboard. Okay, so you're going to take 2 test tubes. One you're going to label HCl for hydrochloric acid, and one that you're going to label NaOH for sodium hydroxide. And you'll put those in your test tube rack. There are 3 other test tubes that you're going to be labeling for activity 2, but you don't need to do that yet. Okay, then you're going to take 2 of the 1 milliliter pipettes and you're going to label those as well. So those 1 milliliter pipettes are the ones that come in your general equipment bag, not in the pH lab bag. Okay, so one of those I've labeled hydrochloric acid and one I've labeled sodium hydroxide. These work by squeezing the bulb and they suck up water when you squeeze the bulb, and they also dispense water. And you can see there are measurements on the side for measuring liquid. Okay, so you label those and set those aside also. I'm just going to put those by my rack. Okay, now you're going to label 4 of the bigger pipettes, and you're going to label those water, pH 4, pH 6, and pH 8. Those come in your pH activity bag today, and you can see that they are fatter in diameter than the ones that came in your general equipment bag that you labeled sodium hydroxide and hydrochloric acid. Those are smaller. So this one I labeled a W for water, and then I've got pH 4, pH 6, and pH 8 that I've labeled. Okay, so I'm going to put those aside. Now you're going to get a beaker from your general kit and label that water. And you're going to add about 100 milliliters of water into this. Again, you can use tap water today. And the reason they say approximately for this part is because you're just going to be using this to dispense water to make your dilutions. So they're saying you need about 100 mils of water. So I used my 100 mil graduated cylinder, and I actually got it precise because I don't like to do anything approximately. And you want to use, to get things precise, you would want to use the one that says W for water, and either add or subtract water until you get right to the 100 mils. You always want to measure with the graduated cylinder, it's more precise, and then pour it into the beaker. The beaker is a good vessel for storing things and for doing an experiment. It's not good for accurate measurement. Okay, so now I have my 100 mils of water. Now comes the calculation part. Number 6 and number 7 ask you to calculate the volume of sodium hydroxide that you would need to prepare 12 milliliters of a 0.1 molar solution of sodium hydroxide. You're going to use the same steps as before. The only difference is now it's 12 milliliters total solution rather than 10 milliliters that were in the example. Okay, so I've done this for you, and I'm going to just try to show it to you right now on camera. Let's see, I'm going to bend this. Okay, so I'm using my equation that I like better, which is molarity 1 times volume 1 equals molarity 2 times volume 2. Okay, so we're starting with a 1 molar solution of sodium hydroxide, and we're going to be diluting it to get it to a 0.1 molar solution. And I want 12 milliliters is going to be my final volume. And if you look at my math, what you do is you rearrange this equation. So obviously we're going to divide by the 1 molar, which if you divide by 1, it's really the same as doing nothing, but it's going to allow us to cross out units. So if you look, I divided by 1 molar, so now moles here cancel out with moles here, and I'm left with milliliters. And you can see that I need 1.2 milliliters of sodium hydroxide to make this solution. Here's the second part of the equation. I'm going to be adding 10.8 mils of water to my 1.2 mils of sodium hydroxide to get the total solution. Okay, I'm actually going to post this calculation on Canvas so you can see this, because it's kind of hard to see right now. You're going to do the same calculation for hydrochloric acid, and it's the same numbers. Okay, now here we go. Add the sodium hydroxide labeled and hydrochloric acid labeled dropper pipettes to the respective test tubes. What that's asking you to do is those skinny ones that you labeled, you're going to put the one that says hydrochloric acid in the hydrochloric acid test tube and the one that says sodium hydroxide in this. You don't ever want to cross-contaminate. So you don't ever want to use the wrong dropper in the wrong test tube. Okay. You also now want to prepare these two solutions. And in order to prepare those two solutions, you're going to have to use the calculation that you had before. So you're going to need your small graduated cylinder. Okay, and what this is telling you is to get 12 mils total of a 0.1 molar hydrochloric acid or sodium hydroxide solution, you're going to need to add, as we calculated, you're going to need to add 1.2 mils of the acid or base and 10.8 mils of water. Really, all you need to do is add the 1.2 mils of the acid or base and then add enough water to get you to 12 mils. Okay, so if I want to add 1.2 milliliters of my acid, what I'm going to do is I'm going to take this that says hydrochloric acid. This is 1 molar hydrochloric acid. What I'm doing right now is number 7. It's the same as number 6. But I want to add enough to get to 1.2 milliliters, which is kind of hard to see on here. Okay, and then I'm going to add enough water to get to 12 milliliters. So I'm going to take my water and I'm going to add enough to get to 12 milliliters. What I'm going to do is I'm going to get to 10, and then I'm going to use the pipette labeled water, and now I'm going to add enough to get me to 12. Okay, now I'm going to take this diluted hydrochloric acid, I'm going to mix it by gently tapping it against my hand. And I'm going to add it to my test tube labeled hydrochloric acid. Okay, I'm going to do the same thing for sodium hydroxide. I'm going to go rinse this out in the sink first. Okay, we don't want to contaminate this. Next solution. Okay, then I'm going to add 1.2 mils of pure sodium hydroxide. You have more sodium hydroxide than hydrochloric acid because this gets used for activity 2 and the hydrochloric acid does not. So you use this for 2 activities, that's why you have more of it. I'm going to add 1.2 mils of that to my graduated cylinder, and then I'm going to add enough water to get me to 12 mils, and I'm going to pour that into the sodium hydroxide test tube. Okay, now what I want to do is I want to add pH indicator to both of my test tubes. Okay, so the pH indicator is this dark solution. It's called Bogan's pH indicator. There are a lot of different pH indicators that are used in science. This one happens to be the Bogan's. What's important to note about this pH indicator is it really only reads pH 4 to 10. So if you end up looking at your pH chart and you see that something is reading at pH 4, you'll actually record that as equal to 4 or less than 4. So less than or equal to 4. Okay, and then the same at 10. If you record a pH 10, that could be actually higher than 10. So you're going to say greater than or equal to 10, greater than or less than 4, if that makes sense, because it can only read 4 to 10, this pH indicator. Okay, so you're going to put the drops of pH indicator in your test tubes and you're going to note the color of that. All of that we just did, that was all just prep. Also in your prep, you need to label your tray. Okay, so there's a picture showing you how to do that. You're going to label 1 through 5 across the top and then A, B, C, D along the side. Each one of these wells is going to end up with something different in it. You also want to make sure that there are no holes in your tray. And I had to go through it and kind of press out the wells because some of them were bent from shipment. So really make sure you go along and really press this in. Make sure you have some really nice well-shapen wells for doing your experiment. Okay, after you've done all of that, now you're ready. Okay, so basically row A is going to be your control row. So across the top here, you're going to end up with just water in your in row A. And row B, you're going to end up with your pH 4 buffer all the way across. Row C is going to have your pH 6 buffer all the way across. And row D is going to have your pH buffer all the way across. I'm not going to walk you through every single step of this, but I do encourage you to highlight and write notes so you understand what you're doing. Remember, at the end, you're going to take a picture to document your results. You're then going to rinse this out because this same thing gets used for part 2. Okay, so activity 2 is going to require you to label 3 test tubes and 3 more of these droppers. Okay, so one of them, the water one you're going to use again, you're going to label these pH 4, 6, and 8, and then you've got your water one. And you're also going to label 3 more test tubes. Okay, so I've been keeping these in the beaker, and I've got them labeled 50%, 25%, and 12.5%. And now those are going to go in my test tube rack, and I'm going to follow the instructions for activity 2. Okay, now that you've done activity 1, activity 2 is not as difficult, but just really follow step by step, really highlight and think about what you're doing, take a picture, and in the questions, you'll be analyzing what all of this means. Remember, there is an activity 3. In activity 3, you're going to just add water to the beaker, pH indicator, mix, take a starting picture of what happens before you blow the bubbles, then you're going to use a straw and you're going to blow bubbles and note the final color. Okay, if you have any questions, please contact me. Again, I'm not going to take you through every step of the experiment because that fun is for you, but really the prep this week takes a lot of time, so be prepared for that. If you end up needing extra time for activity 3 because you don't have a straw, then you could submit your results for 1 and 2 and just save your chemicals and do 3 later. Really for 3, you just need the pH indicator and some water. Okay, thanks. Good luck. Have fun.